Hello everyone. In this video discussion, I will be covering the enzymes. So this is the last topic that we will be covering for our prelims. So the following are our objectives for this discussion. So of course, we will start with defining the terms that involves on the topic enzyme for us to understand its structure and its mechanism. We will be covering as well its physical composition, its classification. So we will be discussing the different factors that may affect the rate of enzymatic reaction, the enzyme kinetics or the movement of our enzymes, including zero order and first order kinetics. And we will be covering as well why the measurement of serum enzyme levels is clinically useful, especially in our field as medical technologists. Okay, so enzymology. So this is the study of enzymes. Okay, so what are the activities of the enzymes? What are the chemical reactions where enzymes is involved? And what is the clinical use of these enzymes? So basically, class, our enzymes, they appear in our serum or the liquid portion of our blood after cellular injury, after there is degradation of the cells, or it can be found as well on the storage areas of our tissues. So that is where the appearance or enzymes can be located. Okay, so enzymes class, they are from a Greek word en, meaning in, enzyme, meaning yeast. Okay, so who among you here are baking? So, di ba that is one of our ingredient, the yeast, in order to allow the sini para mosaka ang ato ang bread. Okay, so because of this yeast, it can increase the cooking time of our bread or the cake. Okay, so long before the scientists they understood the chemical nature of the enzymes, it was usage of yeast were already used in the production of many food that we have, including the alcoholic beverages. So that is one of the component or ingredient of our drinks. So biologic catalysts, so they hasten chemical reaction or speed up chemical reactions that are used to build structures in our cell, or not only build, but they can also be a reason or a catalyst to break the structure of our cells. So they are not consumed during the reaction. They are not undergoing a chemical change after the reaction. So they are um, they are not destroyed. There will be no molecules that will be taken away from the enzyme after the reaction happened. So they are specific in nature, class, and they are essential to physiologic functions in our body, such as the hydration of our carbon dioxide, such as during the muscle contraction, and also during nutrient degradation. Enzyme is very helpful, or it is useful in this type of process. Okay, so we have here illustration for us to understand what is the action of our enzyme. As what I have mentioned earlier, our enzyme, they are specific. So just the same in our illustration here, it is specific. Our enzyme is specific to this type of substrate. So depending on its shape. So if it's the specific molecule, therefore they can create now an enzyme substrate complex, or our ES complex. Okay. So our substrate class, they are like our enzyme, they are specific as well. So our enzyme, they are considered, or they composed this active site. Okay, so this is the region where our substrate attached to. So this active site class, its characteristic, they are often water-free cavity where our substance on which the enzyme acts on. Okay, so I'm gonna clear this one first. Okay, so our enzyme class, they have this allosteric activator. So what is the meaning of this allosteric activator? So this is a place class on our enzyme where a molecule that is not a substrate may bind. Okay, so thus changing the shape of our enzyme, and it can influence the ability of our enzyme to be active. Okay, so while our allosteric binding site, this one allows molecules to be activate or to inhibit, and it can as well turn off the activity of our enzyme. So they will now bind on the site and change the conformation. It can change the shape of our enzyme as well. Okay. So the same on its illustration to further understand our allosteric enzyme. So our allosteric enzyme class, it is an enzyme with two or more protein chains. They can bind on our regulatory sites. So on this area, okay, they can bind on the site in order to change. It can change again the function of our enzyme. They can inhibit or they can start a certain process depending on the function of this allosteric inhibitor. Okay, so for the specificity of our enzymes class, so our enzymes may recognize and catalyze. So it can recognize a single substrate. That they can recognize as well a group of similar substrates, or they can recognize a particular type of bond. Okay, so again, when we say specificity class, it is the ability of our enzyme not to choose exact or what is the fit substrate on our enzyme or on our chemical molecules. Okay. So we have here the types of enzyme specificity. So we have absolute specificity, group specificity, and linkage specificity. So when we say absolute class, they catalyze only one type of reaction for a single substrate. For a group naman, so they catalyze one type of reaction for similar substrates. So it can be multiple substrates with similar function. And the linkage, so one type of reaction for a specific type of bonding. So it must be a group of substances since we are talking about bonding. So when we say bonding, there is a connection of molecules or enzyme molecules. Okay. Okay, so the following are the terms that we may encounter with our discussion today. So substrate, so this one is acted upon our enzyme and they are specific in nature. So we have isoenzyme. So our isoenzyme class, they are different forms of enzymes, but when it comes to function or action, they have the same action. Okay, so they may differ in its size, they may differ on its physical properties, but as to its action, so they can offer the same action on a certain molecule. So we have cofactor as well. So they are non-protein molecule of a conjugated enzyme. So cofactor class, this is very important because some substrate, they might need a cofactor in order for a substrate to attach. So its presence is very essential in order for a chemical reaction to start. So we have two types of cofactor. We have the metal ions or activator. They are inorganic in nature, and we have our coenzyme. They are organic cofactor. Okay. So we have also our apoenzyme. So they are the polypeptide portion of a conjugated enzyme and inactive enzyme. So again, when we say polypeptide, so they are apoenzyme. And then we have our holoenzyme. So this is a combination of our apoenzyme plus our cofactor. So we will be encountering the following terms later on. Okay, so in this illustration class, our we have here our cofactor, we have here our substrate that is specific into our binding site of our enzyme. So we have here our cofactor, which is the copper. So this is a very important or essential part of our enzyme in order for our substrate to attach and will result to a enzyme substrate complex. So that is our cofactor to further understand. So in this illustration class, we have here our combination of our apoenzyme and our coenzyme. It will now result to holoenzyme once this one is already attached and activated. And we have here the presence of our substrate complex. Okay, so this one class will result to a to to catalyze or to speed up a certain chemical function. So it requires the presence of the following in order for an enzyme to function. Okay. Okay, so we have here a table on the different parts of cofactors. So I have mentioned earlier no na katong we have two our metal ions that are inorganic in nature and we have our coenzymes that are organic. So we have another one, we have our prosthetic groups. So they are organic compounds the same with our coenzymes, but they are tightly bound to our apoenzyme. So they are combined or bound to our apoenzyme. Okay, more terms. So we have our proenzyme. So they can be termed as zymogen. So most of this class, they are digestive enzymes and they are secreted from the organ of production in a inactive form. So this type of proenzyme class, they are usually inactive or nearly inactive, but they are converted into, or our proenzyme are converted into active form by the process of proteolysis. So this process class will activate our enzyme. So again, proenzyme in active form, our enzyme is now the activated form through the process of proteolysis. Okay, so we have examples here, class, on the table of our zymogen or inactive enzyme. So we have there our trypsinogen and our chymotrypsinogen. So these types of proenzyme class, they are secreted by the pancreas. So upon secretion, they will travel or transferred to our intestinal tract in order to be activated. So the activated form, it is now called trypsin and our chymotrypsin. Okay, so they must proceed to another tissue or on the same tissue for our proenzyme to be activated. Okay, so for the nomenclature of our enzyme, so as what I have mentioned before, so our enzymes, they end in ACE. Okay, so that is how we can identify if this one is an enzyme or if this one is a protein or amino acid. So for lipid activation, so we have our lipase, ester, esterase, we have our protein or protease. Okay, so we have illustrations here, no, on which we can get these types of proteins. Okay, so protein from food, protease, so they give nutritions in our body in order to activate no, some functions or chemical reactions in our body. Okay, so according to reaction it catalyzes. So we have five reaction. So we have oxidation. So we can see oxidase. So we'll be explaining this one by one later on. We have reduction, reductase, hydrolysis, hydrolase. So that is the nomenclature that we can see on our enzyme. Removal of hydrogen atoms, dehydrogenase, and removal of carboxyl atoms or groups is decarboxylase. Okay, so our enzymes class, they have this EC numbers or this enzyme commission nomenclature numbers. So we can see that each enzyme, they have this EC number. Okay, so this is how they give EC numbers on our enzyme. So the first digit or kining 1, so that is the class. The second digit, that is the subclass. The third and the fourth, so that is the serial number of our enzyme. So later on, we can see no mga EC numbers of our different enzymes. Okay, so according to its classification naman class, so we have 6 oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Okay, the first one is we have here our oxidoreductase. So there is a process of oxidation and reduction. Okay, so for the process of oxidation, there is removal of hydrogen ion, while on the process of reduction, there is acceptance of hydrogen ion. So our EC number for these types of enzyme is 11127. Example is our lactate dehydrogenase. So we have here class an example on how oxidoreductase work. So remember, so there is a removal of hydrogen atoms and acceptance of hydrogen atoms. So from this chemical formula, our lactate dehydrogenase so converted into our lactate through the enzyme lactate dehydrogenase, it will now result result to pyruvate. So as you can see, the hydrogen atoms was now removed from our original or principal molecule, which is the lactate. So this hydrogen molecule on the pyruvate or the result product is now added. Okay. So the next one is our transferases. Okay, so from the name itself, there is transfer. Okay, so unsa man ang gina-transfer ani? So it's our functional groups other than the hydrogen no, that was covered on the oxidoreductase. Okay, so if ang na-transfer is our amino group, so that enzyme can be named as transaminases. If ang na-transfer naman class is the phosphate group, so example, ATP to ADP. So it's kinases. So example of our enzyme, we have our 2-oxaloglutarate aminotransferase. So with the EC number 2.6.1.1, our aspartate aminotransferase. Next is our hydrolases. So it catalyzes hydrolysis. reaction in which the addition of water molecules to a bond causes the bond to break. Okay, so um, we have here examples of enzymes that undergo hydrolysis. So we have carbohydrases, so breaking of glycosidic bonds in oligosaccharides and polysaccharides. So when we say polysaccharides class, they are a carbohydrates that are polymeric in form, and these types of carbohydrates contains long chains of monosaccharides unit. So, okay, the next one is we have our proteases. So, breaking of peptide linkage in our proteins, and our lipases, so breaking of ester linkages in our triglycerides. So, I already mentioned this one when we say triglycerides, they are composed of three fatty acids or lipids. So, example here is our alpha amylase. So, our with an EC number of 3.2.1.1. Okay, so we have our liases. So it is an enzyme that catalyzes the addition of a group to a double bond or the removal of a group to form a double bond in a manner that does not involve hydrolysis or oxidation. So walay presence of water, walay presence of hydrogen ions, okay, or removal of hydrogen ions. So we have examples here, class. We have our dehydratase and we have our hydratase. So when we say dehydratase, so it is removal of the components of water from a double bond, while our hydratase is the addition of water to a double bond. Example, we have our carbonic anhydrase and our citrate lyase. So, yun na siya. Okay, so under our liases class, so we have these specific types of enzymes according to its function. So our decarboxylases, so it catalyzes or increase the process when there is removal of carbon dioxide from the substrate. We have diaminases if it's our NH3, our dehydratase is our if our H2O, and hydratase is the addition of our H2O or our water. Okay, our isomerases naman class. So it is an enzyme that catalyzes the isomeration or the rearrangement of atoms. So there is an interconversion of our geometric or on the optical position of our isomers. So there is rearrangement. So, only one product and one reactant or substrate involved in this type of enzyme. Example of this one class is our phosphoglycerate mutase. Sorry. And then the last one, according to its action, is our ligases. So this one, it is an enzyme that catalyzes the bonding together of two molecules into one with the participation of our ATP. So this one class, it can catalyze the joining or ligases can, this type of enzyme can catalyze by joining two large molecules by forming a new chemical bond in the process. So example of this one class is our DNA transcribed to RNA. So it needs the participation of ATP in the process. Okay, so we have here a table. So this can be found on your book. So just screenshot this one for you to understand more. Okay, so now let's proceed with the enzyme kinetics, or what is the action of enzyme and who created this theory. So it's we have here our Michaelis-Menten theory. So it's Leonor Michaelis and Maud Menten who originated the theory of enzyme kinetics. Okay, so we have here E plus S, so it's enzyme plus our substrate will result to enzyme substrate complex, and the result would be enzyme plus the product. Okay. So they have formulated class two different theories when it comes to enzyme kinetics or the movement of our enzyme. So we have our lock and key and our induced fit theory. So for our lock and key class, so there is just like our regular na mga locks in our houses. So our key that serves as a substrate and our lock that serves as the enzyme. So, it is very specific, 'di ba? If you're going to use a different key that is not specific for the lock, of course, it will not work. Okay? So, very simple lang siya na type of mechanism or kinetics or action of our enzyme. While for our induced fit naman class, so what if our substrate, it has a different shape compared with our active site? So this one class, this type of theory, so this one, our lysozyme or our this enzyme, it will adapt to the size of our substrate or onto the shape of our substrate. Okay. So this will happen, class, no, if there is an increase pathogen invasion or infection on the part of our patient. So the tendency of our body is to adapt with the changes on the substrate, okay, in order to enhance or inhibit a certain activity or chemical reaction in the body. Okay, so more examples for us to further understand these two models or kinetics of our enzyme. Okay, so this is our lock and key. So enzyme plus our substrate, it can result to enzyme substrate complex. And as you can see in this illustration, our enzyme remains unchanged. The shape of our active site remains the same. Okay, while on the induced fit model class, our enzyme will now adapt the size of our substrate. Okay, so the similarities of these two models. So they both of these types of models or kinetics, they have only one substrate, will only work when it meets the active site of the enzyme. So both of these models class, it requires the presence of enzyme and our enzyme, no, of our substrate and enzyme. While its differences, so as what I have mentioned earlier, our induced fit model, na siya ang appearance ni enzyme is now different. So there is changes. While si lock and key, there is no changes on the appearance of its enzyme. Okay. So, factors that can influence enzymatic reactions. Okay, so the first one is we have the concentration of our substrate. Okay, so the rate of a chemical reaction, it can increase as the concentration of our substrate increases as well. Okay, so the enzymes, it can be greatly speed up the rate of our reaction. However, class, when our enzymes become saturated along the way, when the concentration of our substrate is very high, so this can affect the action of our enzyme substrate complex. Okay, so for we have our first order reaction, second order reaction, and the zero order reaction. So let's cover this one by one. So for our first order reaction class, so are those which proceed at a rate exactly proportional to the concentration of one reactant. So this type of reaction class, they are directly proportional to the substrate concentration. So kung increase ang concentration ni substrate, increase pod ang concentration ni enzyme. So that is the substrate concentration. So direct na siya. Okay. So, while for our second order class, so this type of reaction, this is one whose rate is proportional to the square of the concentration of one reactant. As you can see, it requires two reactants in order for a product to occur. So, not only the substrate present, but also the reactant. Okay, so it will depend on the concentration of both of these molecules present. And the third one is we have our zero order reaction. So the reaction are zero order with respect to the reactants. The rate of reaction class depends on the concentration of catalyst or on some factor other than the concentration of the molecular species undergoing reaction. So this type of reaction class, they are independent to the concentration of the reactants. So no matter what unsa kataas ang concentration ng reactant, as long as the same ang concentration ng enzyme, it will go through or follow the concentration of our enzyme. Okay, so the presence of reactants will not affect the concentration or the effectivity of our enzyme. Okay, so the second one is we have our enzyme concentration. So the higher the enzyme level, the faster the reaction will proceed. So that is very self-explanatory. And another factor is we have our pH. Okay, so it is functional between the pH 7 to 8. So neutral to basic ang iyahang desirable na pH class. So changes in pH may denature the enzyme. So but when it comes to selection of pH class, dili man kaayo that as in little ang pH requirement for our enzyme. So usually they have a very broad pH maximum. So some small changes in pH, it can have a little bit of change on its activity. So dili ra kaayo nga ana kadako ang iyang effect. So examples here of our enzymes with its pH requirement. Okay, so when we say optimum pH class, so this is the pH requirement of the following enzyme for it to reach its maximum activity. So for example, our urease, example ang iyang pH is nasa 1. Of course, it cannot reach its maximum activity. It should be maintained within 7.0 or neutral pH. Okay, so another factor is we have here our temperature. So 37 degrees or body temperature, that is the normal active form or active temperature where our enzyme can do its activity. But if it will reach 40 to 50 degrees Celsius, it will start to denature. Okay, so increasing temperature, of course, it will increase the rate of chemical reaction, but if it will go beyond the required temperature, it will undergo denaturation. Okay, so for each 10 degrees Celsius increase class in the maintained temperature, the rate of the reaction will approximately double. Okay, so until of course or it will go beyond the protein or the enzyme will now start its denaturation process. So we have now here our cofactors. Presence of cofactors can affect as well the enzymatic reaction. So if the cofactor as what we have mentioned earlier, cofactors, they are organic in nature. So, non-protein entities that must bind to a particular enzyme before a reaction occurs. So we already defined this one earlier. The presence of cofactor class, this one is a requirement on some of our enzymatic activity to start. So it can be metallic or non-metallic. Okay, another one class is we have our activators. So presence of activators, um, they can they can start the process of oxidation or reduction. So depending on the requirement of our molecule. So they are usually linked on our substrate to the enzyme or on our coenzyme. So it can increase the process of enzymatic activity. And then we have here our inhibitor. So this is the opposite of our activators. So they can interfere with enzyme reaction. Okay, so we have competitive, we have non-competitive, we have uncompetitive, and we have irreversible. So let us discuss this one by one. So inhibitors. When we say competitive class, they they copy the shape of the original substrate, and they will try to link on the active site before the presence of the substrate. So it will block the reaction of the enzyme. Thus, the enzyme will not have its maximum activity. Next is our non-competitive. So our inhibitor um, will not stick or go on the active site. So this one, from the name itself, will not compete with the enzyme activity. Okay, so this one, it cannot speed up the reaction since it attaches to a wrong active site. And for our irreversible inhibitor class, so for this one, it forms a covalent bond to part of the active site permanently. So with the presence of this one, our inhibitor will not be removed permanently. So therefore, as a result, our substrate cannot 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 placed on the active site, and the action of our enzyme cannot be activated. Okay, so more illustration to further understand. So we have our competitive inhibitor, unahan niya si site, si substrate in order to attached on the active site. And then we have our non-competitive. So it will attach on the non-active site, and then we have another example of uncompetitive. It will not alter the reaction or um, um, the the function of our enzyme substrate complex. Okay, so more illustrations to further understand the inhibitors that we have. Okay, so how can we measure the enzyme activity that we have on our concentration? So we can measure this one if we can identify no the concentration of the product. Once taas ang imuhang product, therefore increase pod ang enzyme activity. So if the substrate is decreased, therefore decrease pod ang imuhang product. Okay? So, we can measure as well the decrease or increase na concentration of our coenzyme and an increase in the concentration of the altered enzyme. So, how are we going to measure the following activity? So, what are we going to perform? Okay, so we have here different principles. The first one is we have our coupled enzymatic assay. So this one class, so we can determine the activity of our substrate or enzyme by the process of coupling our enzymatic reaction with another. So therefore, it can be easily detectable if the reaction occurs or if the speed of the reaction is increased. Okay. The next one is our endpoint assay or fixed time assay. So for this process class, there is a time that we need to follow. Now within 1 hour, how many ba ang product or how many ba ang reaction? Okay, so how many reactants are combined? If how many substrate or enzyme complex was created within 1 hour. By that, we can measure the amount of reaction has occurred. Example, after 1 hour, increase ang rate of reaction. Then after 1 hour, another na pod, times 10 ang reaction sa atuang enzyme substrate complex. And then we have our continuous monitoring or kinetic assay. So this is multiple measurements. So we can measure this one on a different times. So there is time interval. So depending on the need of our or on the rate of reaction, it can be after 30 minutes, it can be after 1 hour. So we can measure this one through absorbance. So, if increase ba ang absorbance or decrease ba ang absorbance? So, in this type of assay class, we are using spectrophotometer na equipment. Okay. And the last calculation of enzyme activity. So I know. So these are the units that we can use when measuring the enzyme activity. So we can use international unit or IU, muna siya inyong makitaan ng mga unit of measurement, no? So, amount of enzyme that will catalyze the reaction of 1 micromole of substrate per minute under specified condition. And international unit per liter, IU per L. So enzyme, that is, we are measuring the enzyme concentration. And Catal unit, which is mole over second. So that is the amount of enzyme that will catalyze the reaction of 1 mole substrate per second under a specified condition. Okay, so that is the way of measuring the enzyme activity. So that's the end of our discussion on enzymes. Thank you, everyone, and have a good day.